In the last class, we have studied what the MRI molecular imaging is, how it does, and give some clinical practice. And I think most students may understand the aim of this subject. Yes, we want to achieve the specific and target imaging. Even treatment are precise diagnostics, which may provide accurate evaluation for the diagnosis and treatment. In addition, because we can get the images and perform the treatment for disease at the same time, this technique solves another very important problem in clinical: how to achieve the real-time monitoring for the treatment effects, how to track the migration of the drugs in body, how to know the drug concentration in the region. As you know, all of these are very important problems in clinic, so we should know the importance of this technique. Recent 2 decades, there are so many developments in MR molecular imaging. However, most are still in the preclinical stage, so we should pay more attention to promote the clinical transformation. As we have discussed in the last class, the MR molecular imaging is a medical imaging technique. It can directly display psychological and pathological process of cells or molecular in real and complete human or animal bodies through images. So it may detect the physiological process and pathological mechanism of structure or functional changes of the pathological cell in biological tissue, quantitatively or qualitatively, such as gene expression, metabolic activity level, and intracellular biological activity status. Except the typical MR molecular imaging uses targeted nanoprobe. Actually, there are so many other methods to complete the molecular imaging. For example, the chemical shift imaging. The location and the distribution of some MR visible inherent or exogenous compounds or metabolites which are related with special molecular process in the body can be detected using the chemical shift imaging. It has shown great clinical application value in the diagnosis and evaluation of some diseases. Like this, some other imaging methods such as DWI, SWI, functional MR may be broadly classified as the scope of the molecular imaging. Since they are already widely used in the clinical practice, we will also discuss this method in the class, except the typical target molecular imaging. Here, we will talk about some advances in the MR molecular imaging in the nervous, advanced system, and tumor. Finally, we will discuss the first development in the future, and part 1 is nervous system. At first, we will discuss different MR molecular imaging techniques and its imaging features. Secondly, we will talk about the current problems and the prospects in the future. Using the nanoprobe to achieve the molecular imaging is the classic imaging method. Other suitable molecular imaging techniques today we use include micro MRI and some different imaging sequence consist of MR spectroscopy, MRS diffusion weighted image, and functional MR. The micro MRI is with sensitivity, transmit coil, more effective path sequence, and higher magnetic field intensity from 4.7 Tesla to 14 Tesla. You know, the clinical MR scan today is less than 3 Tesla. Micro MRI is used to scan small animals with higher spatial resolution and isotopic resolution. It can achieve the organic, histological, even cellular imaging. However, the scan time is long in order to get better images and more information. Micro MRI may directly display the metabolism in vivo, collects RNA information, and then reconstructs the images of the protein structure, composition, and metabolic process. For example, microMR can be used to detect amyloid plaque in transgenic mice with Alzheimer's disease in vivo because of the iron deposit in the plaque. T2 star sequences are magnetic sensitive imaging SWI and could be used to clear, show, and count amyloid plaques. On the image, amyloid plaques on the hippocampus or cortex of the mice are shown respectively, and the immunohistochemical stain confirm the close relationship between both. The right image shows the amyloid plaques using the novel liposomal nanoparticles by MRI. If you use the high-resolution SWI, some tiny amyloidosis plaque was shown here, and the detection effect is similar to the fluorescence and the histological staining. In recent years, with the development of the molecular imaging, MRI tracing has been applied more and more in evaluating the efficacy and the safety of the stem cell transplantation, organ transplantation, bacterial infection, gene exploration, and drug development. For example, USPL as marker can be engulfed by bone marrow, mesenchymal stem cells, and the cell will be hyperintensity on T2-weighted image. Thus, we can monitor the treatment efforts of stem cell transplantation in ischemic or degeneration diseases or other diseases. MRS is an important and available sequence to get more information about by molecular and chemical profiles that represent different aspects of the molecular metabolic process and the amount of the specific substance in cells of many nervous diseases. On the MRS image, we find this normal brain with a low cooling peak, an index of cell proliferation, but a higher cooling peak in the patient means active cell proliferation, which suggests a malignancy for the tumor. Another patient shows the similar appearance on MRS. Diffusion-weighted image can show the water molecular diffusion is usually used in clinical to detect the tumor, infection, abscess, and so on. A new water channel protection molecular imaging with metal B values can be used to identify the high and low grades of the brain astrocyte tumors. Besides this, functional MRI is defined as a board or ASL method that can be detected abnormal signal changes in brain cell activity before structural brain damage occurs. Actually, the function MRI in broad senses include 3 parts. The first one is cerebral blood flow measurement consists of contrast enhanced and perfusion and board. The second is MRS, and the third is nerve fiber tracing, including diffusion, tensor, and magnetochemical transfer imaging. Among them, the functional MRI of board effects is the most widely used. Board is blood oxygen level dependent functional MRI. When local brain region is active, the deoxyhemoglobin increased, resulting in the change of T2 signal. This reflects the function of local cerebral cortex active and is most important method to study the brain function today, using the change of the blood oxygen concentration in brain as the natural contrast agent. For example, the image shows MRI area in connecting brain regions has a tendency to reduce, compared with the normal side, suggesting the function damage of this side. Another image reflects the difference in brain function changes between response to the face and to horses. Alzheimer's molecular imaging techniques in this part show process structure and functional information for improvement of the diagnosis and therapy of the nerve diseases. 9. Invisibly. In addition, it can be used to study molecular neurobiological invasion. There are still some problems about MR molecular imaging in nervous system, such as imaging accuracy, sensitivity, resolution, and spatial labeling. And the BBB must be considered when we use the targeted probe. Fortunately, with the rapid development of MRI scanner sequence, hybrid modalities, and more precise model, the molecular imaging in brain will solve more problems in clinic. Okay, this is all for this part. Thank you.